Let's start with our first laboratory session entitled Bacteria Everywhere. Participants will be challenged to hypothesize about where more bacteria are found. They will develop awareness that bacteria are actually everywhere and that various surfaces might have different loads of organisms. The skills learned in this lab will prepare the students for other labs and activities in the food safety curriculum. The first part of the experiment should not require more than 45 minutes. Each student or team should use the student worksheet to enter the areas that they want to explore and their hypothesis as to which have more or less bacteria. Remember to do this safely. It's a good practice to wash your hands with soap and water, then use personal protective equipment such as goggles and latex or nitrile gloves. Seal the culture plates once they're inoculated and never open them because some of the organisms could be dangerous pathogens. We are using pre-poured nutrient agar Petri plates that came in a sleeve. Note that the plates were stored upside down with the gel in the top portion to avoid condensation dripping on the surface of the gel. We will store our inoculated plates in the same manner. Place your first plate on your work surface with the lid down. We are also using sterile saline, which you may have prepared or purchased from the drugstore. I prepared mine and labeled the container to identify the content, the date it was prepared, and the initials of the person who did it. Now let's start. Each team should have 2 or 3 Petri plates and a small container of sterile saline solution. Now we grab a permanent marker to prepare our plate by writing on the bottom. We split this one in 2, one side for our blank and the other for the swab. The result from this plate will tell us if our agar and the saline and swabs are sterile. Of course, don't forget the date and initials. Now grab a sterile swab by the wooden end and dip the cotton tip in the sterile saline solution. The cotton now holds saline and either no bacteria, if it was really sterile, or some bacteria. We'll find out later. To culture the bacteria, we streak our sample onto the half of the plate prepared for the saline. Streaking means to make a zigzag pattern without lifting the swab. As we move, the sample gets deposited onto the gel. It's more concentrated when we start and diluted after a few lines. And now we don't touch the blank side. Okay, so we bring a second plate in our work area, gel side up again, and split it into 4 quadrants and label each for one of the sampling areas entered in the laboratory worksheet, date and initials. We're ready for our samples. Grab a new sterile cotton swab, dip it in saline, and rub it on the surface of your area of interest. I'm using a drain basket that I purposely placed in my kitchen sink for 2 or 3 days without washing it. I used the sink as normal, but did not pick up the basket to wash it. I like to have the grossest sample as part of my demonstration to students. Then we repeat the streaking pattern, but this time a bit smaller to cover only 1 quadrant of our plate. It's now time to sample our surfaces of interest. You may want to look at a toilet, a sink, where I would suggest the drain or faucet handles, or other surfaces such as a doorknob or a cell phone. Each surface is rubbed with a new sterile cotton swab dipped in saline and streaked in the correct quadrant. If you did not completely fill your worksheet before preparing the samples, please go back and complete the table on the left side. Now store all the plates in a dark and warm place for 2 days.